Good afternoon, everyone, and welcome to this webinar hosted by the MRC HTMR Trial Conduct Working Group. I'm Chris Rogers from the Clinical Trials and Evaluation Unit at the University of Bristol, and it's my great pleasure to introduce Louise Bowman, our speaker today. Louise is an Associate Professor at the Clinical Trials Service Unit at the University of Oxford. Her specialist clinical background is in diabetes and lipidology. She's a particular research interest in cardiovascular disease in diabetes and is co-principal investigator for the ASCEND trial that we're going to hear about today. Louise has a particular interest in trials methodology with a focus on approaches to large-scale recruitment strategies and streamlining methods for clinical trials. So without further ado, I will hand over to Louise and she will be able to tell us about the ASCEND trial. Louise.
Thank you very much indeed, and welcome to everybody from me with an initial apology that for those of you that were at the hub, the network meeting in Cambridge, this is going to be largely a rerun of the presentation that I gave there. But I hope that some of you will be new, and obviously there's opportunities for questions at the end of the session. I'm going to spend the next 20 minutes or so just describing to you some of the methods that we use for large-scale recruitment, aiming to do important clinical trials but keeping budget low, yet still getting reliable answers. And this is the example of the ASCEND trial, which is a trial in diabetes that is aiming to answer some really very simple questions. So for people with diabetes who have not yet had a heart attack or stroke, is low-dose aspirin beneficial? Are omega-3 fish oils beneficial? And are these treatments safe? And you'd like to think that by now we might know the answer to these questions. These treatments are not new. Aspirin has been around, well, in the form of willow bark since the days of Hippocrates, and omega-3 fish oils obviously aren't a new idea. But remarkably, the data available to us for the management of our patients with diabetes in these respects is surprisingly limited. And so we felt that these were really important questions which needed robust evidence from clinical trials to answer them so that I could make informed decisions about the management of my patients. But the problem here is, of course, that both of these treatments do not come with heavy backing from the pharmaceutical industry in terms of funding that might be available for running large-scale trials. And the sort of benefits one might expect from these treatments are only ever going to be modest, and so the sort of trial that was needed was going to be large in order to have sufficient statistical power. So we had to think sort of outside the box, really, in terms of how could we design a trial that was going to give us reliable answers and yet still be achievable. So we really had to work on how to streamline our approaches to the trial with a really highly cost-effective methodology. We opted for really simple design, simple eligibility criteria, and in the end chose a male-based trial design to run this trial. And you'll see later on that we've got over 15,000 participants in the study, all in the UK, and the aim is to have 7.5 years of average follow-up by the end of the study, which is actually coming to its close this year. The design of the trial is a 2x2 factorial design, so that's our first bit of streamlining, really. We're doing 2 questions for the price of 1. So in the one arm, we're comparing aspirin with placebo, and then in a separate randomization, we're comparing the omega-3 fatty acid active component with, again, placebo. And it's a really robust way of using your population to answer more than one question, and a design that I think is often overlooked and can be a really valuable tool. The next thing that I'd already mentioned is that it's a male-based study. One of the areas that is particularly expensive when it comes to running clinical trials is the requirement for study clinics and staff. And so for this study, where both treatments did not require any biochemical monitoring, we worked out that we were able to run this trial entirely by post. We could get rid of the need for study clinics, and all we needed was access to a quaint old English postbox available on most street corners, and of course added bonus that that was requiring people with diabetes to go for a stroll out to their postbox in order to post their study questionnaires back to us. Along with that came the challenge of working out how to get study treatments to the participants, and we had to work very hard with the packaging company that we used to design boxes that were going to fit sufficient quantities of study treatment, packaged in a way that met all of the necessary rules and regulations for study drug packaging, and also we needed to make sure that they were child-proofed. So we have a system of pop-out blister packaging, which allows us to provide in the smaller box, that's our run-in box of treatment, which is 2 months of treatment, and the larger box is 6 months of treatment, and these are done in slimline boxes that then fit through the majority of British post boxes. So once we'd sort of worked out how we were going to approach it, we obviously needed to secure some funding, and we were fortunate enough to get a British Heart Foundation special project grant, which has then been renewed on 2 subsequent occasions, giving us a total of £3.7 million from the British Heart Foundation, which I agree is no small amount, but remember, it's a very large-scale trial, and that's covering us for 70,000 person-years of treatment and follow-up. We managed to secure the study drug and funding for the packaging from Bayer, the providers of aspirin, and then Mylan, the company that produced the omega-3 fatty acids. And the trial is coordinated by ourselves at the University of Oxford, it's sponsored by the University of Oxford, and we have an academic-led steering committee and an independent data monitoring committee that oversee the running of the trial day to day. So we've got a design, we've got some drug, we've come up with a plan as to how to deliver this trial. The next problem was, of course, we needed to find our participants. And so we had 2 main routes of doing this. One of the advantages of the trial being in diabetes is, of course, there are registers of patients with diabetes, either hospital-based registers or regional registers, in particular registers that are used for retinopathy screening. And with Section 251 approval, we were able to access these registers without prior consent from the individuals listed in order to invite them to take part in the study. Separate to that, we also had the help of the Primary Care Research Network and the Diabetes Research Network in particular to approach GP practices, and patients were invited directly from their practice to take part in the study. As a separate small print approach, we also had an option for people to just volunteer for the study if they wished, or to send us details of friends or family members that might also be interested, but that didn't really result in very many recruits. So this central recruitment approach involved us identifying potentially eligible patients from registers and then mailing them an invitation letter, an information leaflet, a screening questionnaire, which had the consent form attached to it. And we in Oxford provide a 24-hour telephone service to allow any questions to be dealt with. We have on-call clinicians that cover this service throughout, and in fact it's a system that we've set up many years ago and have always used for our large-scale trials, even when we have clinic-based studies, because we feel that study participants often do have queries that need addressing as a matter of urgency to them. They may not be clinically urgent, but they're important to the participants, and the participants are important to us. And so we provide this service for all of our studies, where they can phone a free phone number and get clinical advice any time of day or night. This slide shows the screening questionnaire for the study. If you imagine this as an A3 sheet of paper, it's folded down the middle, so they receive an A4 sized envelope. The front page is the invitation letter. This is showing the back page there, which is the consent form. And the invitation comes ostensibly from a local investigator at the hospital where the data was obtained. And we have the permissions to get the hospital header, which we scan in to our systems, and signatures of the local investigators, which again we scan in, so that we can generate the very large numbers of these invitations centrally and mail them out without the need for work to be done locally. But it looks like they have come from the local hospital because that's where the data originally resided. This is the other side of the screening questionnaire, and you can see it's very simple, very straightforward. On the left-hand side, we confirm the contact details of the participants and their date of birth, and their GP details. And then on the right-hand side, these are the simple eligibility criteria. Does the patient definitely have diabetes, and then are they known to have had previous cardiovascular disease, which would exclude them from the trial? So simple questions that the participant answers for themselves by ticking the boxes on the form. There are some additional eligibility questions where there is the option for free text as well. And these forms, when they come back to us in Oxford, are then scanned, and we use optical character recognition scanning to help us interpret the forms. So the check boxes are read electronically, and then we have computer programs that highlight areas where there is free text entered on forms, and they can be then diverted to either an administrative or a clinician review process so that the forms can be reviewed and checked and the data processed in a highly efficient manner. Once a participant enters the, if a participant returns a screening questionnaire that demonstrates that they are eligible and they have provided signed consent, they then enter a pre-randomization run-in phase. And during this phase, it's a single-blind part of the study. So we send them a drug pack which has placebo aspirin and placebo fish oils. We don't tell them it's placebo. We know it is. They don't. And this is used as a sort of tester period to check that they really are going to be happy to continue into the long-term phase of the study. And it gives us an opportunity to write to their GP to inform them that they've entered this run-in phase, and also to do a number of other things, including sending out a blood kit to them so that there's the opportunity to provide blood samples if they desire. It's not essential, it's not a required part of the study. So again, in that sort of streamlining way, one thinks about what do we actually need to do to answer those questions that I put on my first slide, and actually we don't need blood samples to decide whether aspirin and fish oils should be routinely prescribed to patients with diabetes. But if we can get blood samples, then that gives us added value in the trial in terms of being able to characterise our population more clearly. So with a separate British Heart Foundation project grant, we got additional funding to provide these kits, which are sent out in the mail with all of the components that are needed to take a blood sample. And we ask participants to see if their GP practice is happy to take a blood sample for them, or alternatively they may be able to get it done at their local hospital if they're known at a clinic there. And then the samples, both blood and urine, are sent back to us again in the post with special packaging that is approved by Royal Mail for the transport of samples. But the entire thing is optional, and actually we had an excellent response to this. We sent out 23,000 blood kits, 13,000 participants returned samples, and of the randomised participants, that was 3/4 of them. And we're able to do a number of baseline biochemical measures on these. Not all analytes are suitable for male samples. So, for example, we can't get plasma creatinine levels. But you can see the list of items there, which gives us a very good baseline characterisation of our population. And we can also store samples long term, including genetic material, which obviously is potentially valuable in the future. This is a picture of our very hard-working administrative team preparing those kits, or some of the kits, which went out on a sort of rolling basis as participants got to that stage in the run-in, 2 weeks into the run-in, when it was the computer systems triggered a request for a kit to be sent out. 6 weeks into the run-in period, they then received their randomisation questionnaire, and here's an example of this, again with a letter on the front, this time coming from myself and Professor Jane Armitage, the joint principal investigators of the study. The back page this time asks participants to tell us about their additional current medication, and again we can use computer systems at our end to code the information that's provided there. We also ask them to tell us about an alternative contact, the name and address of a friend or relative who lives at a different address, so that if they move and they forget to tell us where they've moved to, we've got the option of still getting in touch with people. And then the inside of the randomisation questionnaire firstly checks compliance with the run-in treatment. We only want to randomise people who really have been able to take their run-in treatment reliably. And then a little bit more detail about their diabetes in terms of the history of it, how long they've had it, whether they take insulin, and then confirming their eligibility once again to make sure that nothing has happened during that run-in period that would mean they're no longer eligible. And then finally on that page we have a number of other demographic features, ethnicity, and also ask patients to report their height and weight, again so that we can help to characterise our population a little better. Once again, all of these boxes on the forms are read in electronically with a series of programs that check the data so that we have sort of inbuilt data consistency checks as well. This slide then summarises the recruitment effort, and you can see that through these kind of routes one does have to send a very large number of invitations. Through those central routes, we sent over 300,000 invitations in order to randomise 9,000 patients centrally, which sounds like a large number, but when you have systems that can just automatically generate these on a large scale, you're paying for the cost of the postage, but you're not paying all those staffing costs and clinic room costs. And it is achievable. There are plenty of patients out there with diabetes, so as long as you have a large pool to draw from, it's perfectly possible to recruit these sorts of large numbers. And you can see we got about 3/5 of our patients from the centrally held registers and another 2/5 direct from general practices. With a similar approach where in the GP surgery they electronically screened their own data for potentially eligible patients, told us how many they had. We then provided them with screening packs that contained the questionnaire and the information sheet and all of the other items, and they simply then popped in a front cover letter from the GP surgery where they'd mail merged the patients' names and addresses and put them in the post on our behalf. And then the participants reply back to us in Oxford with their completed forms. This is the geographical distribution of the randomised participants by postcode, and one of the joys of doing a study by mail is you really can reach the parts that other studies can't necessarily reach, up and down the country, with no need for participants to be able to get to a specific study clinic. So quite nice distribution there. Notable that, of course, if you think of the large built-up areas, London features really very poorly, and our experience from other trials is that recruitment in London is very hard, and the same is true in this study. So we're now in the follow-up phase of the study, or just coming towards the end of it, and again, questionnaires are sent every 6 months where we ask participants to tell us about serious adverse events, significant bleeding, we want to know if they've been taking their study treatments, and certain non-study treatments such as aspirin and warfarin that we're particularly interested in. And alongside that, they get their treatment packs mailed out as and when they're required, and we also have all the participants flagged through central registries for death and cancer and indeed hospital episode statistics. And throughout, as I mentioned before, we have our 24-hour free phone service for any queries that may arise either from participants, their families or their own doctors. Just for completeness, this is the follow-up questionnaire, and here we have similar questions about compliance. If participants want to stop taking the study treatments, we ask them to tell us why. Other medications of interest at the bottom of the left-hand side, and then on the right-hand side, specific serious adverse events that are that feature in our primary endpoint. So in particular, we ask participants if they have experienced a heart attack, a stroke, a TIA, all such things, and then cancers and bleeding. And again, we ask for additional information there that we can code directly through the electronic systems that are reading in these questionnaires back in Oxford. And the back of the form there, any other hospitalizations, again, we record details. During the course of the study, we've also developed methods for web-based follow-up. Obviously, internet connectivity has increased apace over the last decade since the study started, and so we now have the option for participants to complete their follow-up information directly online, should they wish. And this gives us the advantage that we can have a number of checks built into the form electronically, so that we don't have missing fields or inconsistent fields. So if a participant wants to do this, they get sent a link to the form that's unique to them, so that it's obviously associated with their study ID, and they simply click to start the form, and then it's the same questions that you saw on the paper version. How regularly have they been taking the treatments? And here's an example of somebody missing out the first answer, and it comes up, if you try to proceed, it comes up with a query saying please make sure you've answered all of the questions, so that we don't have missing data. One of the difficulties of doing a mail-based study is, of course, one misses out on some of that face-to-face interaction, which is incredibly valuable. In a long-term study such as this, the need to keep people motivated can be problematic. So we have made efforts to produce regular newsletters, which we send out en masse to the study population, and those are very well received. We try to cover any particularly relevant stories. So, for example, over the course of the send, the interest of aspirin in relation to cancer has been quite notable. So we'll often pick up on news stories and help to provide more information about those in the newsletter. So to summarise, we used these mail-based methods to recruit 15,000 participants by mail. It's one of the largest ever studies in diabetes. And I think the setup of the NHS really helped with that. It might not be achievable in other countries. We've used a very streamlined trial design, very simple case report forms, where the participants complete the information themselves, no study clinics at all. We're in the final phase now, so participants are currently being sent their final follow-up form, and it'll take us probably most of this year to gather all of that final information. So we're anticipating that the results will be available in 2018. It's a very simple, straightforward study, but with over 400 million people in the world with diabetes, the results of it actually will be extremely relevant to those large numbers. And we hope that from a public health point of view, it'll be a very important study. If you're interested in the methods that we used, we have published a bit more detail about that. Here's the front page of that publication in Trials from last year. And our website, which is listed there, does have more information. If you just Google Ascend Diabetes, it comes up as the top hit because the website name isn't terribly memorable. But it's very easy to find us with a lot more information there. Or please do feel free to email me if you've got any specific queries. But that's all I was going to say, and I think we've probably got a little bit of time for some questions if anybody has any. Thank you very much.
Yeah. Thank you, Louise. That was a really interesting presentation, and lots for us involved in trial delivery to think about in our own studies. As Louise said, we now do have some time for some questions. If you've a functional microphone or a telephone connection and wish to speak directly to Louise, please follow the instructions on the screen to raise your hand. Alternatively, you can type your question in the Q&A panel and I'll read them out on your behalf. I don't think we have any questions that have come in yet, so maybe I can just start the questioning session this afternoon. And I wondered if you could tell us a little bit more, Louise, that without face-to-face study visits, as you said, it's a very efficient trial design, but do you have issues with retention in such a long-term trial?
Yes, it's very relevant because it's all very well recruiting patients, but actually it's only any good if you can hang on to them. And we all know that in longer-term studies, retention it can be a real problem, and of course your statistical power drops off appallingly if you fail to actually get your study data. So it's an area that we spend a lot of time on in Ascend. The newsletters are one of the aspects that I think really do help with retention, and indeed our free phone line I think is vital in terms of providing participants with the reassurance that they can get hold of us at any time of day and night should they have any concerns about the study. Overall, we're rather pleased with our retention. In the context of such a large study that has been going on for an average now of 7.5 years, if we look at follow-up information available for any individual participant, we have about 95% of the whole study population. We've got complete follow-up information within the last 12 months. And obviously, a real focus of our efforts as the study comes to a close is now going to be ensuring that we do have that final piece of information from all participants. In some cases where it's no longer possible for participants to complete questionnaires themselves, they may nominate a relative or carer to complete the questionnaire, or sometimes even that isn't possible, in which case we then turn to the GP for information, and we have a system of GP follow-up where we mail an abbreviated version of the questionnaire, and obviously these are all going to be people who are no longer taking the study treatment. We can mail the questionnaire to the GP practice, and actually we have a very good response there from helpful practices providing us with the limited information that we need about those key primary endpoints.
Okay. We've got a few questions that have come in now, quite a few in fact. So the first question from Morag McLean is, how do you actually organise the cover for your 24-hour phone service?
So we run a number of large-scale trials here at CTSU, and we have I don't know how many we've got at the moment, 8 or 9 at the moment, I think, research fellows or other clinicians who are happy to man that service. I've been doing it for the 15 years that I've been here at CTSU, and it waxes and wanes in terms of how busy it is. When you're in the recruitment phase of a study, you can get quite a few calls out of hours, but actually at the moment it's incredibly quiet. And it's, we have a bleep rotor and a pager that gets passed around on a weekly basis to cover for a week. And amongst the clinicians, we're all very happy to do that because we do really feel it's a very valuable part of the trial. In terms of the logistics of it, we have a free phone number that comes into CTSU during working hours, and then we're fortunate enough to be able to use the local hospital switchboard system, so we transfer the free phone number over to the hospital switchboard out of hours, and then they will bleep us if a query comes through.
Okay. Before I read out some of the other questions, I know we've got somebody who's raised their hand, so let's go over to Eleanor Mitchell.
I've got a couple of, can you hear me?
Yes.
Yep.
Oh, okay. It was fun, started bleeping. I've got 2 questions, if I may. Firstly, I was just interested in the sending of the letter out centrally, but with the local details on it. I just wondered if that had generated any queries from the participants directly to the site, and how kind of the engagement of the site in answering any potential queries from participants, whether there were any issues there.
Okay, no, that's a good question. Interestingly, there were very few queries. We were very careful to be clear about the contact details and to have our contact details, phone numbers, email addresses, all the rest of it very prominent on the letters, and simply to have the name of the local hospital trust on the header, but not the contact details. Obviously, each hospital that provided us with hospital-based data had agreed to the process. We had local R&D approval for that, and we had a local investigator who was aware, fully aware of the study, had been trained in the protocol and knew what it was all about. But they were simply a participant identification centre, and so once the recruitment phase had completed, their involvement then was no longer required. But it was surprisingly few queries that ever got directed to the local hospitals, I think because we were particularly careful about our contact details.
Okay, thank you. And then with something just very quickly separately, to do with the reporting of safety data. So presumably SAEs were self-reported by the participant, and I just wondered what your process then was for kind of clinical review and onward reporting to MHRA, etc.
Yes, so the SAEs are reported directly, although if a participant was admitted to hospital, quite often we would get a call from their managing team because we provided them all with a sort of membership card, if you like, a sort of credit card sized a small little plastic card that they were asked to keep with them at all times. It had our free phone number on, so that if there were ever any queries, we would be contacted. And so we certainly did get notified of occasions when participants were hospitalised, particularly in the case of, for example, serious bleeding episodes, which one might consider in relation to aspirin. We could then have a conversation with their managing team about that and make decisions in light of the clinical presentation and other clinical factors about whether there was any requirement to report related events. In terms of other events that came in directly from questionnaires that participants report, we then will gather supporting documents from the GPs about endpoints of interest. So the major vascular events, myocardial infarction, stroke, TIA, the elements of our primary endpoint, plus also serious bleeding episodes. We write to the GP and ask them to send us discharge summaries and information about that event so that we can then adjudicate those events according to pre-specified criteria. So all the usual systems really for safety reporting are in place, but it's a case that the participant tells us directly rather than having the intermediary of a study clinic.
Okay, thank you.
Thanks. There's quite a few questions still in the list, Louise, so I'll try and sort of share a couple with you, but I think we'll probably need to aim for quite brief answers just in the interest of time. The first questioner asks, what system of reminders are being used for each questionnaire stage?
So we send out 2 routine reminders, 1 at a month, 1 at 2 months, although we can vary that depending on the stage in the study. And then if we don't get an answer to the second reminder, we'll then follow that up with a telephone call and potentially calls to the alternative contacts if we're unable to get hold of the participant.
Okay. I think you've mentioned about sort of your retention and losses to follow-up, which another listener asked about. So another question was, you use section 251 to gain access to some registers, and Kendra Cook asks, why did you not use section 251 to gain access to the GP registers and then to conduct the mailing from the research team?
That's a very good question. The difficulty is that the data resides at the GP practice, and in order to send the questionnaires centrally we would need to have an extract of data from every single practice, which amounted to something like 700 practices, I think, in the end, did the exercise for us. And it's so straightforward for a practice to do their own electronic search and to mail merge the details in. We felt that we were unlikely to get approval through Section 251 for that aspect. The point of 251 is that really you're not supposed to have any other alternative but what you're asking for in terms of data without consent, and in this instance there was an alternative approach which was acceptable and achievable.
Okay. A couple of sort of technical questions now. The first being, what computer system did you use for reading the CRF forms? And the second, how much trial and error did you have in feeding the forms to be electronically read?
Oh, those are good questions and sound like people that have tried doing this before. We use a system called Eyes and Hands, although we are very fortunate to have a lot of programming support here in Oxford. Part of our approach to these very large-scale trials is that we are very IT heavy, very administratively heavy as well, because we develop a lot of in-house systems to run our trials. So we've used Eyes and Hands as the sort of off-the-shelf package for the actual scanning software, but then our IT guys have bolted on a front end and back end to that, which satisfies our purposes in terms of the reviewing system. So not easy to offer completely off the shelf for the whole process. Trial and error for it, it wasn't easy, I have to admit. We've learnt an awful lot over the years about the scanning process. It is fiddly, it is very temperamental. The forms have to be set up exceedingly carefully. Every single print run of the forms has to be rechecked within the system, so it's not a small undertaking. Again, for a study of this scale, we felt it was worthwhile because there are hundreds of thousands of questionnaires that have been processed through the system now. But for a smaller scale study, I'm not sure if one would necessarily go down that route.
Okay. And I'm just going to pick a final couple of questions on a slightly different theme, and I apologise to the others, and maybe you'll be able to contact Louise directly, just in the interest of time. And these two questions are around sort of, firstly, did you have any complaints from people about receiving unsolicited invitation to join the trial? And sort of in a semi-related sense, as you collect alternative contact information, have you encountered any obstacles from the REC as to collect personal information from people who do not consent to that? It seems to be a problem nowadays, particularly in stroke trials.
Yeah, so in terms of complaints, we certainly did, during the recruitment phase, have people who phoned us concerned as to how we'd got hold of their information and worried that it was inappropriate. And actually, in far and away the majority of cases, when we explained the setup of the trial, we explained all of our approvals and Section 251 and all of that, mostly people were then perfectly happy and generally went ahead and joined the study. We had a small number, I can't off the top of my head remember, but it's something like 20 or so out of those 300,000 who asked us to remove their details from our databases and completely anonymise their record, which of course we did according to all the data protection requirements. So remarkably low numbers in the context of such a big exercise, and often really a simple explanation on the phone was more than enough to satisfy any concerns. In terms of other contact details, it was on the form from the outset that was submitted to the REC for approval, and of course it's the participants that are sending us that information. One would hope that they have checked with that person first that that's okay to do. When we receive that data back, when we get the form with other contact details back, we simply scan the image of that section of the form, but we don't translate all of that into electronic data. So those names and addresses aren't stored in a format in our database that is sort of readily searchable en masse. It's simply as an image for an individual form, so that then we can refer back to that should we need to contact them. And we haven't had any difficulties in relation to that process.
Okay, thank you, Louise. I think we best stop the questions there now, and there are a couple that we didn't have time to ask explicitly, and I hope those people will feel free to contact Louise directly. Just a couple of points before we finally wrap up. Firstly, to say that today's webinar has been recorded, including the Q&A session, and we'll be emailing a link to you within the next week or so about that, and feel free to share that with your colleagues. There is also a questionnaire as you log out from the webinar today. Please do fill it in, because we're very interested in your feedback, and particularly if anyone is willing to give a webinar in the future, we would be very pleased to hear from you. We have a full program of webinars for 2017. The next one will be at the end of March, when we'll be welcoming Rachel Briley, who's going to talk about methods to assess adherence in a cardiac surgery trial. We're not quite sure of the final details for April, but we have Sean Truick on our books for May. So please look out for those. They'll be advertised through the usual channels. And I think it just leaves me then to say, again, thank you to Louise for a really interesting seminar. As the number of questions showed, it was clearly something that appealed to and found relevant to everybody on the call. And thank you all for joining. And so at that point, I'll say goodbye, and I hope to speak to you again next time.